Fink. And actually what I'm going to be talking about today, I will read the title of the talk for everybody. It is intrastratal transplantation of adenovirus-generated induced pluripotent stem cells for treating neurological and for treating neuropathological and functional deficits in a rodent model of Huntington's disease. And this work is really a part of an international collaboration that I've been lucky enough to be a part of between Central Michigan University and the University of Nantes, where we were actually creating and characterizing these cells at both places, and I've had the opportunity to travel back and forth to do this. And I'd like to just point out at the beginning, we'll hit them in the acknowledgments as well, but all of the authors on this paper who really made it possible to complete this this study that we're very excited about. So briefly, I'm going to go over a little bit about Huntington's disease, a little bit of the background on Huntington's disease. Actually, move this and kind of move to the side, I think. And a little bit of the previous work, a little of the background of how we got to where we are right now at Central Michigan University using adult stem cells to treat these neurodegenerative disorders, specifically Huntington's disease. And I'll cover what our work is with mesenchymal stem cells, some of the mechanistic actions that we see with our mesenchymal stem cells, why we think they're providing functional recovery. And then I'll move in and try to spend the majority of the time actually talking about these induced pluripotent stem cells, the work that we've done with those both in France and at Central Michigan University. Some of the take-home messages, and again, the acknowledgments. So what I'd like to start with is just a brief summary on what is Huntington's disease. Huntington's disease is an autosomal dominant disorder. It's characterized by cognitive and motoric deficits, usually occurring in midlife, that progress with, at death about 15 to 20 years after the onset of the motor symptoms. It's characterized, as you can see here, this is a healthy brain over here on the right, Huntington's disease on the left, and we see these medium spiny neurons here in like the caudate nucleus here, undergo massive degeneration and massive cell loss. We also see thinning cortical regions, as well as lateral ventricle enlargement. And this is really a myriad. Huntington's disease is a global disease. Every cell actually carries this genetic mutation, but the areas that we focus on mostly are these basal ganglia structures, and what we'll refer to in the animal models as the striatum. And as of right now, the only FDA-approved treatments for Huntington's disease actually focus on just suppressing the movement disorder. And outside of a couple of fetal transplantation into the brain of these humans, these Huntington's disease patients, there's really been no successful way to slow the progression of the disease. And with these fetal transplantations, although we have got a mixed result of how well they can actually slow or prevent the onset of these symptoms, they're fraught with all kinds of ethical issues of availability, immunological issues, as well as tumor formation. And so really searching for another type of stem cell that may be able to help in this disease is really vital to moving forward in research. And so in our lab, in 2003, they published a paper where they transplanted whole bone marrow into a toxic lesion model of Huntington's disease. And what they observed in this study is, it's one of the first studies to really show, these are the animals that just received the toxin, our healthy animals here, and then the animals that received treatment. This is a radial arm maze. And we can see over time, in the number of errors, that the animals that actually received this autologous bone marrow actually had a reduction in the number of working memory errors. So this was very exciting. This is one of the first times that we've been able to see a functional effect of adult stem cell transplantation in a Huntington's disease model. But interestingly enough, because when we think stem cells, we think cell replacement, creating new neurons. But this wasn't the case. They were able to find the transplanted cells. You can see here that they're metabolically active, they're still alive, they're still in the system. But they didn't become neurons. And so they started to speculate, what are the possible mechanisms of why these cells are exerting this functional benefit without becoming new neurons? And they started to hypothesize that maybe it's due to trophic support. And so the next study that we did was, again, transplanting a purified form of these mesenchymal stem cells into another toxic lesion model. And we can see here, in the 3-nitropropionic acid model, we have a large lesion. And in the groups here, and we can see in lesion area, the ones that receive transplantation of stem cells have a reduction in the size of this lesion, as well as that, from that first Huntington slide, you could see we had an enlargement of the ventricles. And here we also see another reduction in the enlargement of the ventricles following transplantation of the stem cell. But we were speculating at this point that it was due to trophic support. And that's actually what it turned out to be, is these mesenchymal stem cells were releasing a myriad of different trophic factors, but the most important one that we feel in Huntington's disease is this brain-derived neurotrophic factor. So our mesenchymal stem cells are in here, they're releasing brain-derived neurotrophic factor, and we're getting these functional effects. And so we thought, again, in this lab, how can we can expand this and push this to see even a greater effect. And so what we did with these mesenchymal stem cells is we genetically engineered them to actually overexpress brain-derived neurotrophic factor. And when we did this, we transplanted them into a transgenic model of Huntington's disease. And here, this is a Accelerod task. And so the longer they are, the longer they're capable of remaining on this rotating rod, the more we can speculate that they have intact motor and coordination. And we can see down here in the black, these are Huntington's mice, and you can see that they're doing poorly throughout the duration of the study. But when we have these genetically engineered mesenchymal stem cells, we can see it almost normalizes their performance. And so we're really seeing that these mesenchymal stem cells that secrete these trophic factors are very vital in Huntington's disease. We also saw here, interestingly enough, in our untreated YAC128 mice, compared to our wild type, our non-transgenic mice, we see a significant loss of medium spiny neurons, and these are the neurons that are very selectively lost in Huntington's disease. And following transplantation of our BDNF mesenchymal stem cells, we see that this is almost restored, prevented or restored. And so these mesenchymal stem cells are helping slow the progression of the disease, but again, they're not becoming new neurons. And so what we think, too, is another thing that may be going on is not trophic support, but also immunomodulation of these cells, because we know we have inflammation inside of the Huntington's brain. And so outside of just trophic support, what are these cells doing? And so we conducted another study with Dr. Rossignol, where we wanted to look at the immunological or immunomodulatory properties of these mesenchymal stem cells. And so we co-transplanted them. In this study here, we can see our mesenchymal stem cells, and then we co-transplanted them with adult neuronal stem cells. Adult neuronal stem cells should have the ability to differentiate into neurons following transplantation. Although in our experience and in some other labs, we see that they are subjected, these adult neuronal stem cells are subjected to a large immune response and eventual rejection from the host. And so we thought if we had these mesenchymal stem cells that can create a better microenvironment, release these trophic factors, as well as create maybe a micro immune suppression in the striatum for these other cells to survive, these adult stem cells would have a more efficacy for treating the disease. And interestingly enough, we found that yes, our mesenchymal stem cells do modulate the immune system, and that they do allow these more sensitive, these cells that are typically rejected, to be able to survive. And here we can see this is just the area of the transplant. And so what we did in this was we just delineated the limitations of our transplant. And we see that our mesenchymal stem cells have great survival there. They're always around, they survive very well, very robust, and we can always find them in the brain following transplantation. We see that our adult neuronal stem cells have about, and the same number of cells, so 400,000 cells were transplanted per hemisphere in this group, and then 200,000 mesenchymal stem cells, 200,000 adult neuronal stem cells were transplanted in the co-group. And so we can see that our adult neuronal stem cells had a significant rejection when they were transplanted alone, that we couldn't find them, that the transplant area was significantly smaller. But following, if we co-transplanted them, so the same amount of cells here, we actually see that this group has an intact graft, that we can find these, that the mesenchymal stem cells are allowing them to survive. But we questioned whether or not this is just the mesenchymal stem cells being the mesenchymal stem cells and having a large graft area, or is it specifically with the adult neuronal stem cells too? And what we did when we actually did cell counting of these transplanted sites here, we saw that the adult neuronal stem cells had very few surviving cells inside, and when we counted only the adult neuronal stem cells in the co-transplantation group, we saw a significant increase in the number of surviving cells. So our mesenchymal stem cells are not only providing trophic support that is beneficial to these Huntington's disease mice, but they're also providing a local immunomodulatory effect where we can transplant other cells and help push the differentiation of cells or help increase the survivability of these cells. So a quick summary, this is a lot of the background work that's was done previously that I've participated in the lab to get to the point where we're at right now, and really the presentation that I'm going to be giving you about the induced pluripotent stem cells. And so the main take-home messages with mesenchymal stem cells are that they provide trophic support. They release BDNF. They also release some NGF, some GDNFs, and different things. But for Huntington's disease, the one that we're most interested in is the brain-derived neurotrophic factor. We also know that it creates a better microenvironment inside of the striatum or wherever this transplant is. And we think it's due to immunomodulation. And this immunomodulation is a release of specific interleukins and cytokines that actually kind of block the infiltration of lymphocytes or T cells or macrophages. And we think that we can use that to help create a better environment that we can transplant cells into and not have to worry as much about rejection, and also being able to avoid global immunosuppression with like cyclosporine A, which we know has a myriad of side effects and may limit the clinical utility of these cells. However, the big take-home message, well, not the big take-home message for this talk, is that we didn't see them become new neurons. So why these mesenchymal stem cells are working wonderfully, we're seeing trophic support, we're seeing behavioral sparing, we're seeing a reduction in the cell loss, we're seeing a sparing in the neuropathological changes typically observed in Huntington's disease, but they're not becoming new neurons. And that was a big issue for us because we thought really, I mean, during the progression of this disease, you have lost cells, and we want to be able to create new neurons to replace those cells that were lost. And so the question now is cell replacement possible? We want to replace the neurons that are lost in this disease. And if it's applicable in Huntington's disease, theoretically you could use these cells for Parkinson's disease, traumatic brain injury, stroke. If you can create a way to get neurons that will be able to be transplantable, survive in the brain, differentiate into the correct lineage, and hopefully provide some efficacy. So what we're moving into now is the induced pluripotent stem cell realm. And what induced pluripotent stem cells are, so these are adult somatic stem cells, skin cells, hair cells, dental pulp cells that we can expose to 4 different transcription factors. And if we can force these 4 different transcription factors, and these 4 transcription factors are present during embryogenesis and then are silenced during adulthood. So if we can force these 4 factors, we can actually see that we can reprogram the cells back to an embryonic-like state. And if you guys are keeping up on the Nobel Prize, Yamanaka in 2012 for his pioneering work that just got published in Nature in 2006, so this is very rapid to get a Nobel Prize. But his pioneering work of actually being able to show, and they're referred to now as the Yamanaka factors, and I'll talk about it in a minute, that we can actually take adult stem cells, push them back to an embryonic-like state, and now these cells have the ability to differentiate into any lineage, so you can use them for all kinds of different applications. And then Sir John B. Gurdon, who was vital in actually almost 50 years ago now, with somatic cell transfer, and actually also confirming that you can reprogram cells in a different way, although Yamanaka really revolutionized the efficiency and the viability of these cells after you reprogram. And so this is a very dense slide, I'm not going to go through everything. But over here on the left, this is normal differentiation. And so you can start with a naive pluripotency. And so this is really like a blastocyte cell. So as soon as the zygote's formed and you have these initial cells forming in the body, and these cells are the ones that have unlimited self-renewal, leukemia inhibitory factor dependent, and they have the ability to become every cell in the body. And you can actually isolate these cells and expand them out. These are what are true embryonic stem cells. And then just during normal development, they become these embryonic cells. They lose this naive pluripotency. They have a downregulation of these pluripotent genes, OCT4, SOX2 nanon. They continue down the path of differentiation. They start to lose the prime potency. They start to become germ layer specific, so now they're becoming more what we're considering multipotent cells like adult neuronal stem cells that differentiate into neurons, glial cells, oligodendrocytes, but they're never going to become, outside of cancer or tumor formation, they're never going to become skin, they're never going to become bone. And then they become these early lineage cells, and then at the last step is we get into a differentiated state. So typically these cells are senescent. They're only going to express surface expression markers specific to that lineage of cell. And then they're not going to express any of these pluripotent things. They're pretty much stuck where they're at. And so what Yamanaka showed is if we express or force the expression of this OCT4, SOX2, KLF4, and c-MYC through viruses, plasmids, small molecules, peptides, different things, we can actually force the cell backwards. And you actually see the cells go through these different intermediate stages where they take on factors of multipotency, and then these early pluripotent cells are what a lot of people are calling pre-iPSCs, where they look like an iPSC, they express things like an iPSC, but they're not fully capable of differentiating to every lineage, until eventually you can push them all the way back past this early potency stage where you still have some methylation of the host cell down here into a true induced pluripotent stem cell. So this cell is almost identical to an embryonic cell. and that it can differentiate into every lineage of the body. And it's really exciting that we have this. And so some of the potentials for using induced pluripotent stem cells is you can take a patient's cells, you can do a skin biopsy, you can expand these cells in culture, expose them to these transcription factors, and make an induced pluripotent stem cell. So it's patient-specific. So you can take somebody with a rare disease, isolate a skin cell from them, push it into an induced pluripotent stem cell, and then from that induced pluripotent stem cell, you have a disease state cell. You can differentiate into a neuron, a skin cell, a muscle cell, whatever you need to. And you can use it for disease modeling. And so, like I said, you can differentiate these cells into any type of cell that you want to, be it skeletal muscle, bone, glial cells, brain cells, medium spiny neurons. And then you can use it to screen drugs. And so drugs that are made specifically for this patient's cells can be tested on the specific lineage of cell. And then that drug should be able to go back to the patient. That's an alternative to using embryonic, so these true ESC embryonic stem cells or fetal cells for transplantation studies. And so we can take these cells, these patient-specific cells, and then we can take them back to the patient, either differentiating them into a lineage that we're interested in transplanting, or we can just take them and transplant them back into the patient. And so when you're generating these induced pluripotent stem cells, and I've mentioned this or alluded to this a few times, there's many ways to do it. Two of the more famous ways, and what Yamanaka initially published, was using a retrovirus, which is going to permanently integrate these 4 factors into the cell. And when these 4 factors are forced, you get an induced pluripotent stem cell. Except the drawback of this is it has a high efficiency. That's not the drawback. The drawback is that c-Myc and KLF4 are implicated in oncogenesis in cancer cells. And so even though people suggest that once you get an iPSC, these factors, or the exogenous factors, are actually silenced, that you still have these inside, and it also can disrupt the karyotype, some of the genetic modeling of the cells. And so there's another way that we can do it, and that's with an adenovirus, which is only going to transiently express these, not permanently integrate, but we'll be forcing the transcription long enough to actually force it into an induced pluripotent stem cell. And so some of the work that we've done, we've used mesenchymal stem cells with an adenovirus, mesenchymal stem cells with a single cassette lentivirus, so we can compare, up here and down here, these 2 methods. And we see that our reprogramming efficiency, it looks really low, but we're actually fairly content with having even this many cells formed. And then we also took tail-tip fibroblasts with a lentivirus and tail-tip fibroblasts with an adenovirus, and we see that they, although form less colonies, we still get iPSC colony formation. And this stuff was just accepted for publication in Cell Transplantation. Hopefully we'll be having this out on PubMed soon, and so you can read about some of the different characterizations of these and the differences between the reprogramming methods and what we feel are really the best way. And I'll touch on that a little bit in this talk. And so how we did this with our induced pluripotent stem cells is we expand our fibroblasts, we expose them to the virus, we get these beautiful little iPSC colonies that form. Not a lot of them, but we get some. And then we go in and we actually hand-pick these. We take a sterile pipette under aseptic techniques, and we pick these cells, and then we expand them onto a layer of deactivated feeder cells. And you can see that these colonies start to grow and form. And then when we look, we can see that these cells express NANOG, TRAL160, stage-specific embryonic antigen 3, and stage-specific embryonic antigen 4, which are all markers of pluripotency. And so we see that we can start with a skin cell, which would actually be over here. I took that picture out. We can expose them to these factors, get an induced pluripotent stem cell, expand these on feeder cells, and we see that they have at least the surface expression markers of a pluripotent stem cell. When we look at this with RT-PCR for the mRNA of these cells, and so this again are all the different groups from that paper I said that we just had coming out. So we can see our lentivirus, our tail-tip fibroblasts and mesenchymal stem cells, as well as with our adenovirus, again in black are our tail-tip fibroblasts and then our MSCs. We see that both of them do express OCT4, which is a marker of pluripotency. Our adenovirus cells actually express more. We see a similar pattern in our KLF4 with our TTFAD, or tail-tip fibroblast adenovirus reprogrammed cells, expressing the highest amount of these pluripotent markers. SOX2, we see that we get expression of both of these pluripotent genes, and with c-Myc. And so this is the endogenous c-Myc, which still is somewhat of a concern, but we think this is normal in our induced pluripotent stem cells. From this data, we kind of gathered that our tail-tip fibroblasts with adenovirus, and so this group here, is actually our most clinically viable, or most, the cells with the most utility. What we think are the most clinical utility, as tail-tip fibroblasts are easy to obtain, We're using an adenovirus, which is non-integrative, and so we don't sit in different things. And so we took these cells, well actually on the next slide here, we tried to differentiate all these cells into neuronal lineages. We can see, so these are just our different groups here, our mesenchymal stem cells with lentivirus, our mesenchymal stem cells with an adenovirus, teltic fibroblast lentivirus, teltic fibroblast with an adenovirus. And we can see with NCAM and nestin here, with NCAM is a marker of maturing neurons, and we see that all of our lineages, when we put them into this differentiation protocol, are capable of differentiating into these neural rosettes expressing NCAM. And a little bit of nestin, we see that there's really no big differences between our groups, but they're all significantly higher than our, or that's our nestin, but they're all significantly higher than our control cells, our teltic fibroblasts that weren't reprogrammed. And so we think that these cells, at least in vitro, have the capability to differentiate into neurons, which was pretty exciting for us. So the next thing that we did is we actually took these cells and we transplanted them into healthy rats. And when we transplanted them into healthy rats, we did a time course study, and so we wanted to look at both the immune response to these cells, if the cells could survive, and then if they would differentiate following transplantation. So here you can see this is confocal images in the striatum of our transplant. The induced pluripotent stem cells are in blue. We pre-labeled them prior to transplantation. In green is our mature neurons, NUN, and then in red, this is DART32. And what we can see at all time points in black are our transplanted iPSCs is we have a significantly robust survival in these healthy animals. And when we look in terms of neuronal differentiation, so we're looking again at the co-localization here. Here in this confocal, you can see on the sandwich view here, we can see the blue neurons surrounded by NUN and DART32 on the surface. And so these cells are beginning to differentiate into mature neuronal lineages, not only mature neuronal lineages, but medium spiny neuronal lineages. And we can see in black, these are our transplants at different time points, 5 days, 21 days, 63 days, and 90 days. For neurons, we see that, and this is percent of transplanted cells. So we get cells that transplant or differentiate into neurons, but again, low. And so we started to think about ways of how we can explain this, and we were excited about the region-specific differentiation, but again, the efficacy is low, or the percent of differentiation is low. And so we started to speculate, well, is this due to the environment that we're putting them in? And so the next study that we wanted to do is, okay, we're sure that the cells can survive. The cells are differentiating into mature neurons, into region-specific neurons, but these are healthy animals, unilateral transplantations, and so we have no behavioral efficacy, we have no functionality of the cells. And so what we did was we transplanted them into a Huntington's disease rat model. And how we created this model is through the administration of 3-nitropropionic acid. So 3-nitropropionic acid is a neurotoxin that can be injected systemically, and it's going to specifically target those medium spiny neurons in the striatum. Another interesting point that we were interested in this study was that the therapeutic time points. And so when you're actually transplanting these cells, we know Huntington's disease is a progressive disorder. And so the loss of neurons happens over many, many years. And in the 3-nitropropionic acid model, we actually do this in the administration in a dose-dependent way. And so they progressively lose neurons. They progressively show these behavioral deficits. And so we wanted to look at the time points of, okay, if somebody, because we can genetically test, we know when they have Huntington's disease, is it better to transplant them at the very earliest time point we can, before they start showing symptoms, before we see this robust cell loss? Is it better to transplant in the middle stages of the disease? Is it better to wait until most of the neurons are already gone and then try to actually go in and replace those? And so we wanted to examine that in this study. And then we did a behavioral analysis, a very basic motoric task with these animals, the acceleron. Then we did a histological analysis where we looked at loss of metabolically active tissue in the striatum. We looked at structural changes in the brain. We looked at the immune response to these cells. We looked at different structures, higher magnification structures inside the brain, as well as the neuronal differentiation, which again is a big point that we'd like to make. And so this is our behavioral data. And so here, again, this is an acceleron task. This is the latency to fall, how long they can actually maintain balance and coordination on this rotating rod. That speeds up. It goes from 4 RPM to 40 RPM over the course of 2 minutes. And we look over time. So baselines before they received any 3-nitropropionic acid. At week 1, we can see, so our gray group here is our animals that were only injected with PBS, so no toxin, and we see that they can maintain motor coordination throughout the duration of the study. Up to 10 weeks is what the endpoint that we had in this study. We can see the black group, these are the animals that were injected only with 3-nitropropionic acid, delivered IP. And we can see that at baseline, they're fine, and then as we begin to give them 3-nitropropionic acid, we see a steady progressive loss of motor coordination that's remaining throughout the study. And interestingly enough, in blue here, so this is our early transplantation group. This is 7 days following the administration of 3-nitropropionic acid, so they got transplanted right here. We see that at the beginning they show a drop in motor performance. But then, this blue group, as we go along, is actually able to maintain. You see a slight upward trend here, it's not significant. But there are several points here where we see that our animals that received induced pluripotent stem cell transplantations are significantly better than our untreated animals. When we look at our intermediate transplantation group here, so at 21 days in red, we see that they follow that same pattern. They get transplantations, and they begin to maintain at several points where they're also significantly better than our untreated 3-nitropropionic acid animals. So we're seeing some functional sparing with these groups. And then one of the most interesting groups to me, and we're still trying to parse out exactly what everything means when you'll see the rest of the data, is our 42, so our late-stage transplantation. We see that they go and they decline, and they're terrible here. They're right on par with our untreated 3-nitropropionic acid animals. They receive transplantations of induced pluripotent stem cells, and we see that they come back up. Actually, right here, where they're significantly better than our untreated animals. And then we have some transient points here and here. And this is really the first time that I feel comfortable talking about functional recovery. We see that the animals are getting better following iPSC transplantation. Then we look at some of the structural changes inside the brain, and so the metabolically active tissue using cytochrome oxidase in the striatum. And we can see here in our untreated 3NP animals and our late-stage transplanted animals, we see a significant decrease in the amount of metabolically active tissue, or what was positively labeled with cytochrome oxidase. And we see that our early and intermediate transplantation points, that loss is prevented. When we look at the area of the lateral ventricle, we see a significant increase in our 3NP group, and this is prevented, or actually we see a decrease in our lateral ventricles and in our transplantation time points. So then we want to look at the immunological response to these cells, and with IgA1 for activated microglia, we can see, so this is optical densitometry of microglia, and so we see in all the groups that received 3-nitropropionic acid, we had an increase in activated microglia in the striatum, and that was increased in our animals receiving transplantation, so we don't think that these cells are immunomodulatory. They're not suppressing the immune system. When we look at IgA1 for macrophages, we see that we have a lesion effect. Our 3NP animals have significantly more macrophages in the striatum than the rest of our groups, and then the rest of these groups we don't see that same infiltration. When we look at the GFP, so astrocytes, in the transplantation, because we have typically following these renal striatal transplantations, we have an activation of the astrocytes, and we can see that we don't have a lesion effect between our sham animals and our 3-nitropropionic acid animals, but we have a trend as these animals had transplantation very early, middle, and late. We see an increase in the timeline. So these animals only had the cells, or these animals had the cells for the shortest amount of time. They also had the largest amount of astrocytes, maybe due to the surgery being closer to the end stages in the other groups. And so again, these cells are not really doing immunomodulation. And then when we thought, okay, let's look at trophic support. So we took the brains, we isolated the striatum, isolated the RNA, the mRNA, we looked at BDNF, and we see that we don't have a lesion effect, we don't have any differences in brain-derived neurotrophic factors. So our cells aren't just pumping in BDNF. So the mechanism of how their functioning is different. When we look at tumor necrosis factor alpha, which is a kind of a global inflammatory gene, we also don't see any changes. We see in this group they were a little bit less than our sham and our 3NP, but we don't think that these cells are really suppressing the immune system. Their mechanism of function is not due to immunosuppression or immunomodulation. And so interestingly, when we started to look at the transplants, and so we have our sham, our early, middle, and late stage transplantations, we can see our iPSCs were relabeled with Hoche again, and then our 3NP animals alone. So some of the first things that we looked at was just, are the cells there? Are the cells surviving? And interestingly enough, what we found was that our early transplantation group, so at 7 days, actually had the most surviving cells out of the other two groups, indicating that possibly transplanting these cells into a a more preserved brain would increase their functionality, increase the survivability of these cells. Another thing that we looked at in terms of structure, as you can see here, these are striasomes. These are cerebral spinal fluid-filled vesicles in the striatum. You see they're usually uniform and nice size. In Huntington's disease and in this toxic lesion model, you can see that they're greatly increased. And so we measured these. We looked at these, and what we saw is, again, we had a lesion effect with our 3MP animals having significantly larger striasomes than our sham group. And then in our early transplantation groups, we see that these are, or the enlargement of the striasomes is actually prevented. In our late stage, we also have large striosomes. So the late stage transplants, why that was able to prevent some of these motor deficits, or actually cause a restoration of the motor deficits, we're not seeing neuropathological sparing or restoration of the neuropathological changes that occur in this model. And then quickly we'll go through these. We looked at the number of mature neurons using NUN, and we see a nice lesion effect between our 3MP animals and our sham animals. And this was prevented in the animals receiving early transplantation. And when we look at the amount of induced pluripotent stem cells that are co-localizing with NUN, we see that these animals actually have the highest percentage compared to the other two groups of iPSCs that co-localize with NUN. So these cells are differentiating into neurons in a degenerating striatum. When we looked at the number of medium spiny neurons using DART32, a very specific marker for medium spiny neurons, again we see the same loss, lesion effect, loss of cells, but this is prevented in all transplantation groups. These are more transitory points than a complete restoration above the 3MP group. And when we look at the co-localization, we see that all of our transplantation groups, again, were capable of differentiating into medium spiny neurons. And so in quick summary of this study, we saw that transplantations of induced pluripotent stem cells were able to cause a functional recovery, a functional motor recovery in this rat model of Huntington's disease. And if you remember that that late stage transplantation group, we actually saw the group's performance actually trend up. And that the transplantation, especially at the early time points of 7 and 21 days, were able to preserve these anatomical features and slow the histological deficits associated with Huntington's disease. And one of the most important points, or one of the most interesting points I think for me, was that these cells were actually able to differentiate in a toxic environment into these region-specific medium spiny neurons. It was very exciting for us moving forward into different disease models and different tasks. And then again, to kind of tie everything together, there may be a potential for the combinational therapy using mesenchymal stem cells and induced pluripotent stem cells. Some of the big take-home messages overall: adult stem cells can significantly reduce some of these Huntington's-like deficits. MSCs offer trophic support and immunomodulation. They're very important to study. They're very important to use as an application to help in other transplantation studies or as a treatment as themselves, because they can be genetically reprogrammed and transplanted, and they appear to be safe. And that induced pluripotent stem cells may be able to help regenerate or replace the lost neurons. And so we don't know if this is a prevention happening, a replacement happening, a sparing, a preservation, and this is some things that we're exploring. And then overall, some of the big take-home message, if you slept through the rest of the talk, that adult stem cells are an exciting avenue for clinical transplantation. A lot of people I'd like to thank. Most of them are sitting back over there on that side over there. So big thanks to them, specifically Dr. Gary Dunbar, my advisor at Central Michigan. Dr. Laurent Lescadron was my advisor during my time at the Inserum Lab at the University of Nantes. A postdoc when I started, but now as a faculty member at CMU. Dr. Rossignol, he's also sitting back there now. He's our new counselor for CMU. Ming Lu, who was vital in the creation of the actual adenoviruses, quantifying these, helping me get the cells reprogrammed. A graduate student that's helped me a lot, Andrew Crane. All the members of the Inserum Lab, and one that I'd specifically like to highlight is Dr. Xavier Levesque, who actually was in the lab with me in France, and then he just liked hanging out with me so much that he came to CMU with me, and then we continued to work on these induced pluripotent stem cells. His work focusing on Parkinson's disease, hopefully you saw his poster. Today, some very exciting stuff using these cells in a different disease model. The Chateaubriand Fellowship allowed me to travel back and forth. The Field Neuroscience Institute for Restorative Neurology, which is a big part of the funding that went into this. And so at this time, I'd like to open it up for any questions, concerns, insights. Thank you.
we have time for a couple questions?
I'm curious as to what the 3NT actually did to GABA levels in either one of the models. And if there was a change, maybe one was predicted, could you restore that?
In terms of GABA loss with 3-nitropropionic acid, GABA levels. You would expect in 3-nitropropionic acid to have a loss of the GABA, and other labs have reported that. That's not something that we've specifically looked into yet, but I think will be a big part of what we're doing moving forward. The mRNA, the quantitative PCR data that we put up with the BDNF and the trophic support was really our first step. But we're interested in exploring some of these different transmitter levels, protein expression levels. So yeah, that should be something that we're looking at very soon, and hopefully can share with that maybe next year's meeting. Is there any other questions? All right, thank you.